Welcome to this special presentation from the Asia-Pacific Coral Reef Symposium 2021. In this talk, you'll hear from Dr. Lindsey Deignan, who led a fascinating project exploring how coral microbiomes vary across different reef sites in Singapore, offering insights into how corals may adapt to environmental stress. This was one of the first research projects I was involved in during my time at CELSI, where I began my journey into coral microbial ecology. Hey guys, this is Aaron Low and Wrong from Design to Discover. Be sure to stay till the end to view the acknowledgement slide and the full team behind this important work. And if you enjoy science-based content like this, don't forget to like the video, share it with others, and subscribe to support the channel. Let's dive right in.
For the last few years, I've been examining coral microbiomes on Singapore's urban reefs, and today I'm going to share with you some of what we have learned about coral microbiomes from these intensely impacted environments. As we all know, coral reefs are economically and ecologically important ecosystems, yet they are experiencing worldwide declines due to a number of factors. In many locations, coral reefs experience intense local pressures, including water quality issues, disease, overfishing, habitat destruction. However, the largest stressor impacting all corals is climate change, and it will influence all corals regardless of the localized stressors that they face. Not only that, but its impact can be exacerbated by the localized stressors that reefs are experiencing. So my work takes place on the urban reefs of Singapore, where localized stressors are incredibly intense. Singapore is located at the base of the Malaysian Peninsula, at the confluence of the Malacca Strait and the South China Sea. This location has contributed to Singapore growing into one of the most important and busy shipping ports globally. The reefs in Singapore are primarily fringing reefs around the southern islands, just below the mainland of Singapore, adjacent to the shipping port. The proximity of Singapore's reefs to the urban hub of Singapore, as well as its shipping port, means that this system is highly impacted. Another somewhat unique feature of this system is that over the years, through land reclamation, the mainland of Singapore has grown by approximately 25%. This obviously did a lot to reduce the total reef area that was previously present, and it has also made sedimentation a major issue. This, combined with low light penetration, has resulted in a vertical compression of Singapore's reef, which now only occur in the shallow areas, typically above 10 meters. So overall, Singapore's reefs are characterized by intense anthropogenic stressors. Yet, despite all of these impacts, Singapore's reefs persist, demonstrating a sometimes surprising degree of resilience. This resilience is what makes studying the reefs here so interesting. There is a kind of natural experiment set up. We can perhaps see a window into the future where greater anthropogenic impacts are the norm. In particular, examining coral microbiomes in these systems may provide a better understanding of coral resilience, which will hopefully help us to develop more translational tools for enhancing coral resilience. In these studies, we've been focusing on the prokaryotic members of the coral microbiomes. So in particular, we're interested in what characteristics describe corals from highly impacted reefs. To what extent does the microbiome respond to the environment? Then finally, are these microbial communities contributing an adaptive advantage to the corals? One thing that we've learned about the coral microbiome so far is that there are spatial differences in the microbiome community compositions across Singapore. Having established these differences, we wondered then what does this mean for the long and short-term trends of microbiome dynamics, and could this be a source for adaptive resilience? So to examine this further, we conducted a reciprocal transplantation experiment between two reefs within Singapore, Raffles and Kusu, where the microbiomes had previously been shown to be distinct. For this we used Passalopera acuta. In total, we fragmented 6 colonies at each site. After a 72-hour acclimation period, we transplanted half of the fragments from each site to their reciprocal reef. Then we sampled resident and transplanted fragments within each reef 1, 2, 4, and 10 days post-transplantation. Following collection, coral samples were immediately stored in a dry shipper for transport back to the lab, where they were eventually processed for 16S rRNA amplicon sequencing. Like previous studies, we found that the coral microbiomes in this study were significantly different between the two reefs. The corals were dominated overall by proteobacteria and cyanobacteria, with a greater proportion of proteobacteria at Raffles and a relatively greater proportion of cyanobacteria at Kusu. Even though the level of cyanobacteria was lower at Raffles compared to Kusu, it was higher than reported in previous studies. So we suspect that this might be related to the fact that the nitrate was elevated at Raffles compared to the Kusu for the duration of the study. Even though Kusu is closer to the mainland of Singapore, and you would think it would be more exposed to anthropogenic impacts. Another factor that is likely influencing the microbiomes between the two sites is that this study was conducted in April during the intermonsoon period, which occurs from late March to May. And during this period, the directional flow of seawater currents is not as intense as usual. And we typically experience light breezes and hot weather. And it's true, seawater temperatures at both sites were around 30 degrees for the duration of the experiment. So these more hot and stable weather conditions could be why we didn't see as much divergence in the microbial communities between the two sites as was reported in previous studies, keeping in mind that they were still significantly different. And this could also help to explain why there was more cyanobacteria at Raffles, where it was previously not reported in such a high abundance. Coral microbiomes in the transplanted fragments responded quickly, becoming similar to their transplanted reef. It was particularly evident in the two dominant phyla, the proteobacteria and the cyanobacteria. So fragments that were transplanted from Kusu to Raffles, those listed here as transplant KR, shifted to have a higher proportion of proteobacteria, similar to the resident Raffles fragments. While the fragments transplanted from Raffles to Kusu, those transplant KRK, saw an increase in their relative proportion of cyanobacteria. And this shift in microbiome community composition happened quickly. So after just one day, transplants were no longer differentiating from their transplanted reef. Interestingly, we found differences in the microbiome response to transplantation between sites. The microbiome shifted more quickly for fragments transplanted to Raffles compared to those transplanted to Kusu. At Raffles, the resident and transplanted fragments were not differentiated at our first sampling time point on day 1. However, at Kusu, that shift didn't appear until day 2. One factor that may have contributed to this is that the coral samples from Raffles, as well as the transplants from Kusu to Raffles, displayed an increased beta diversity, or greater dispersion, than fragments transplanted to Kusu. So corals originating from Raffles may have taken longer to become similar to the resident Kusu corals because the Raffles microbiomes were comprised of a more divergent microbial community. So conversely, the transition in microbiomes of corals that were transplanted from Kusu to Raffles may have happened more quickly because the samples from Kusu were a less divergent community that became more dispersed. In addition to the shifts in the coral microbiomes following transplantation, there were also temporal shifts in the resident coral microbiomes within each reef. However, the timing of the shifts were not the same for each reef, and we were unable to link the shift in the microbiome with any of the specific environmental parameters that we measured. So this suggests that the coral microbiomes are naturally somewhat dynamic in nature. These shifts could be partially explained by shifts in the relative proportion of the dominant phyla. For example, the shift in microbiomes on day 4, particularly at Raffles, is likely the result of a higher relative abundance of proteobacteria versus cyanobacteria that was observed for that day. Another feature of the microbiome that we identified in this study was a very limited core. So if we defined the core microbiome as being ASVs, or amplicon sequence variants, that were present in 90% or more of all coral samples, then we only identified 8 ASVs that fit that criteria. If we loosened that a little bit to expand it to 70% of all coral fragments, then we increased to only 15 ASVs. So what we have learned from this work and our other ongoing efforts to characterize coral microbiomes from impacted reefs is that they vary spatially across Singapore, and long-term monitoring that we've been conducting also indicates that this spatial divergence is maintained throughout the year. Right? They are temporally dynamic, right? Even over short time periods like days. They respond rapidly to their environment, be that a change in their location or a targeted stress like competition with algae, right? And they can readily fluctuate without succumbing to dysbiosis or death. We believe these characteristics support Singapore corals as good candidates for bioengineering or microbiome manipulation experiments. And indeed, some of our preliminary inoculation studies are confirming this to be true. Given the high-stress environments of Singapore, these corals provide a great opportunity for examining the relationship between the coral host, specific microbial symbionts, and adaptation or resilience to environmental stress. There is still a lot to learn about how the microbiome can enhance host health. However, understanding coral microbiome dynamics may provide a tool for enhancing coral resilience, particularly on impacted urban reefs. I would like to acknowledge the following collaborators for their involvement in this research program. And if you are interested in a more detailed write-up of the methods and results of our reciprocal transplantation experiment, our manuscript was recently accepted by Microbial Ecology and should be available soon. Finally, I'll close by sharing a save the date for the Asia Pacific Coral Reef Symposium, which will be happening in Singapore in July 2023. Thank you.